Morning, Professor.
Good morning.
Oh, Professor, I thought you might appreciate this, but one of the dentists I work with he's a specialist. He does root canals, and whenever you do a root canal, you're taking out all of like the nerve and the blood supply of the tooth, so eventually it becomes kind of brittle. And he's going to go off and do some research on how to use stem cells to restore that blood supply.
Oh wow.
Yeah.
Interesting. But you do know that there probably we didn't cover, but in our textbook there is description of tooth stem cells.
Yeah. I didn't know that.
Yeah. Read the textbook. There's tooth stem cells there. Maybe you can introduce that into the research as well, because if you are in a clinic and you have a chance to isolate those cells and do some research.
For sure, that'd be pretty cool.
Yeah, but especially when you extract teeth.
Yeah.
That's material naturally available to you.
I know. It's a long way. Good
to know that.
Yeah, no, I think that's a pretty big deal.
Yeah. I'm glad to hear that. Yeah, and also, you can put some of your knowledge learned here.
Yeah, exactly. That's what I was thinking.
That's really cool.
Yeah.
Okay, so I will get the class started. So as you know, we will only have 3 classes left, including today's. On Wednesday, I'm going to do an overview of the class. So basically, I'm going to tell you what contents are most important, in terms of the final examination. What should be your focus? I will narrow the numbers of lectures you should focus to about a third to 50% of all the lectures I have given through the course. And also, I will also let you know the key concept. Most likely, I will test heavily in the final exam. And if we still have time, maybe I will give you some practice problems. I don't think we have problem to do many, but a few, like 5 or so. So you get a feel of how the exam will be, given to you. What concepts are more, how I'm going to test those concepts.
Have you decided how many questions the exam is going to be, Professor?
Well, we have 3 hours, so I feel like I'm going to have some matching, matching 2 columns matching, and multiple choices, and some short answers, maybe 4 or 5 short answers. Okay. About 20 or so multiple choices and some matches. Okay. It's not going to be very heavy, so I assume if you attended all the lectures, if you reviewed after class, you should be able to complete the examination within 40-50 minutes at most.
Oh, sounds good.
Okay, yeah, it's going to be short, but if you haven't done what I said, like reviewing, and then it may take much longer. Okay, so we're going to have another student-led discussion on Friday. That's going to be the last one. Next week, we don't have any lecture. Next Friday, it's going to be the final exam date on 11 o'clock, just like our regular class, but it will be 3 hours. So I say here again, it's going to be May 7th, Friday at 11 o'clock. It's going to be, of course, through Blackboard. So I'm going to use the Lockdown browser as well. So if you, I think at this point you must have been familiar with that system. If not, download the browser and get yourself familiar with that. If you have trouble, we can fix the problem right there on the exam date, because we have 3 hours. That's good to have a long time, for elective class. We have sufficient to solve the problems. So today, I welcome you to participate as much as you can, because the topic is going to be very light. We're going to talk about the ethical, economical, philosophical aspect of stem cell research and stem cell as an industry and as a clinical new approach. So feel free, because even if you have learned not much about the technical part of stem cell biology, I feel like you can still participate without any problem today. First of all, it's the ethical part. Not everybody agree that stem cell research is valuable. Some think that it is against their moral standard. So here I give you a bar graph here showing the opinion from people from 3 countries: United States, Canada, and the UK. The blue color indicated that people feel that stem cell research is morally acceptable, while the red color means the percentage of people who feel that it is wrong, it is morally wrong. You shouldn't be doing stem cell research. Apparently, when people talk about the stem cell research, the starting point is their view about the status of embryo. People apparently have a different opinion about what is life. That they have a different opinion. We're going to talk about that in a little bit. But here, as you can see, good thing is the majority of people feel like their stem cell research is morally acceptable. Although in this country, the rate is a little bit lower than Canada and UK, and there are more people who said no to stem cell research. The reason is because, I don't know whether it is related to what group do you belong to, like political parties, Democrat versus Republican. That does play a role, because Republican, generally speaking, more conservative about stem cell research, while the Democrat are a little bit more, forgiving, or they welcome stem cell research. That's just generally speaking. Of course, you can't just simply divide along the party line, because even within the same party, people have different opinions. And of course, that opinion is hugely affected by it's hugely affected by their religion. And here, as we can see, if you divided the people to Catholic, Christian, without religion, and other religion than Catholic and Christian, you can see that both Catholic and Christian have about the similar percentage in terms of supporting stem cell research. Here, there are about 26% of them. When they get surveyed with the question, do you think stem cell research, should you fund the stem cell research? And 26% support that. Well, the majority don't support that. But if you talk about people without religion or with other religion, their opinion are quite similar. About 49 to 50% of people agree that we should support stem cell research. And I guess that has something to do with their view about the status of embryo. Some people feel like as long as it is a fertilized egg, it is the beginning of life. Then you can't touch it. Otherwise, you're destroying an embryo. Some people are very strict about that idea. Well, some have are less restrictive about that. For example, like they feel 14 days is the cutoff. If the age of the embryo is less than 14 days, then probably it's okay for you to touch the embryo and do something, because 14 days is the date when an embryo will be implanted. And before that, it is just fertilized egg. It is if it's in the fallopian tube, it is still floating, right? It's moving, but not embedded to the mother womb yet. Not yet. And some even have a more forgiving opinion about what is the embryo. They feel at 4 weeks, that's when the embryo will develop nerves. They will feel pain, right? They feel like at that time you cannot touch the embryo, and before that it's just a plant, right? They don't have their sense yet. So before that, you probably can do some research with the embryo. You see people have different opinions. They have different view about what is the embryo. So that could be used to explain. Some feel like you definitely cannot do stem cell research. Some will be more forgiving about stem cell research. Then on this slide, what I'm going to show you is what happens in Europe. What's the policy there in terms of supporting stem cell research. So if this green color, of course, that means very permissive. You see a few countries, including United Kingdom, Belgium, and Sweden. Those three countries, they have very permissive rules. For example, like UK, it's probably one of the most permissive countries which have a rule over stem cell research. Some countries just don't have rule at all. So for those which have rules, UK is the most permissive. If you read the first paragraph there, you can use. So by the way, UK is the first country to use in vitro fertilization. So the first human being born from in vitro fertilization was, I believe it was 1978. That happened in UK. So since then, different countries start to apply rules toward embryonic research, not only stem cells, toward how you treat embryos. So UK started early, and then they are more permissive than other countries. As you can see here, of course, naturally, if you start to use in vitro fertilization, there will be surplus, like the sperma and the oocyte not used. And now, in a lot of countries, you can only use those surplus that's already fertilized but not implanted, because you got to have backup. Usually you do quite a few, and then you only implant maybe a couple or one, and then you have a lot of fertilized egg left there, and you can allow that to develop. And then, for human embryonic stem cell extraction, you don't have to implant that. Just in vitro, you will be able to extract the cells. You just allow the embryo to develop for a few days, and then from there you can extract human embryonic stem cells. Of course, they allow you to use the surplus, but they even allow you to create embryos, even if it is not a surplus. Like the scientists want to study some disease, and they want to create some embryos. So the couple can actually reproduce. They don't have any problem. But in UK, it is allowed. You can take the sperm and oocyte and do in vitro fertilization, and then develop embryos just for the purpose of scientific research. That is allowed in UK. That's a big step. Not necessarily available in other countries. And then you see here, they even allow something that's kind of, in my opinion, start to become dangerous, is hybrid embryos created from animal and human gametes. That means you use a human nuclei and use animal oocyte to do reprogram, somatic nuclear transfer. So that is allowed in UK. But there is, another thing I want to mention is even chimeric human embryos. That means you can inject human embryonic stem cells into human embryos, and they go even as far as inject animal cells into human embryos. Isn't that far? It is quite far. But there is a restriction. You can't allow the embryo to develop beyond 14 days. So that means you can never implant what I just talked about. The embryos develop those ways. You can't implant. You can only do scientific research, in a test tube. You're not allowed to move further. Okay. Yes.
Why would they do the hybrid embryos created from animal and human gametes? Like, what would they try to show by doing that?
Well, I think they're not, of course, they're not trying to create a monster, to create a hybrid between human and other species. And most times, I guess they are trying to, for example, regeneration. If you know some species have better regeneration and a human have not as good regeneration of certain lineages, for example, ectoderm. Neuron regeneration is very poor in humans, but in other species, maybe they have more powerful regeneration, and maybe you do that hybrid to see what will happen. Can that bring the human cells to better regeneration? Something like that. Gotcha. Or you can study the regeneration in a humanized environment.
Okay.
Right? That's just something that come to my mind.
Yeah.
Yeah, there could be some other reasons that people would do that, but of course you can even manufacture organs. That could be a possibility. But at this point, the limit is to 14 days. You can't do much. It's only early development.
I do have a question. So what kind of limitations does that pose? Because I know when you get to it takes less than 14 days to get to the blastocyst, right? And that's kind of where the embryonic stem cells are isolated. So does that really pose much limitation on the research that they want to do? That 14
the next stage is called epiblast. So the blastocyst and epiblast, but at epiblast, that's where the implantation will occur.
I see.
So not long after the stage you extract embryonic stem cell, you will implant. So that's 14 days is about the time that you have stem cells, but they don't allow you to go any further than that.
Okay.
Maybe slightly differentiated. The epiblast don't even have 3 germ layers. The next stage is called gastrulation or gastrula. The gastrula will have 3 germ layers.
3 germ layers, yes. Okay.
Yeah, so around that time, you probably have the early stage of 3 germ layers. So that's it.
Okay.
Can't move.
So you don't think it really affects much of the research that they're really focused on then, right?
Yeah, I think that for the most part, you can manipulate the embryo, like we discussed the 3 parents, nuclear somatic cell nuclear transfer to fix mitochondrial diseases.
That's right.
That's something that happened in UK, not here. Yeah, they allow that to be in clinic. Wow. But that those manipulation is within 14 days. It's in an in vitro fertilization clinic.
All right then. So they still have room to do quite a lot.
Yeah. They still have room to do a lot, and especially in disease modeling, they can do a lot. They can create an embryo.
Professor.
Yes.
Like just as we're speaking right now, the news came out saying that the Americans made the first human monkey embryo, which now is sparking concerns to some ethical experts. Right now. Yeah,
I guess that is also for the purpose of generating tissues, something like that. You inject human embryonic stem cells to a monkey, and then you can manufacture some human organs in monkeys. Yeah, I guess that's the reason they're doing that. Yeah, you can have your opinion whether that should be allowed or not. Everybody can have your opinion about that. Yep. So you see, UK, Sweden, and Belgium, they have the most permissive policy, but Germany, Italy, and some other countries, especially Germany, Italy, and Croatia, I believe, they have the most restrictive. At one point, Germany was very permissive, but later on something happened and they start to tighten up their policy, and now it has one of the most restrictive. So how about this country? ever since the first in vitro fertilization was successful, that's 1978, people in this country start to get alerted. They feel like, wow, we need to build some policy to regulate this. This is dangerous. Now you can manipulate embryos in vitro. You can then introduce something or remove something, give treatment to the embryo. It is very, very dangerous. So they start to have policy since that time. So basically, no embryonic research is allowed in this country in early days, since the introduction of in vitro fertilization. But when Bill Clinton took office and he started to push for embryonic stem cell research, in 1995, he proposed to the Congress some changes, but he was blocked. But then in 1998, that's when human embryonic stem cells were first established by University of Wisconsin. It's Thompson first described the technique to generate human embryonic stem cells. But of course, that research is funded by private companies, not by the government, because at that time, government won't support any human embryonic stem cell research. But people start to become very enthusiastic about the possibility of stem cells, right? Generating tissues, fixing problems that at this point we can't do anything about. So Bill Clinton got a very good chance. So he used that, and in 1999, I believe, he was able to push that through the Congress, and all of a sudden, human embryonic stem cell research is allowed since that time. But only 2 years later, and the office was taken by George W. Bush, and he's very conservative about that, and he just passed another law says now we can't do human embryonic stem cell research. We cannot support human embryonic research. It's funny, at some point when you want to do human embryonic stem cell research, the government give you a list. They say that these cells are developed before 2001. And this you can use, because for a window of 2 to 3 years, it is allowed. Therefore, the cells developed with the support by the government, you can use those cells. However, anything that was developed later, you can only do that with private support. The government won't support that kind of research. So for us, when you write a proposal, you need to be very careful. You need to know whether that cell is developed before 2001 or later than 2001. Because if you're not careful, well, your research will not be funded. You spend a lot of time writing, design, experiment, just because you're using a cell line that's not on that list, then you lose the opportunity. So that's what happened in this country. And here I have a plot showing the people's reaction after Bush said that we won't be supporting human embryonic stem cell research. And here is the people's public approval or disapproval of President Bush's decision. You see that there is a lot of disapproval at that point, but it didn't matter. It won't change the outcome. And here is a map. The map shows what the local government like. As you can see, a lot of those Democrat states, such as like California, and then some New England states, as well as New York, New Jersey, and I believe that's Illinois and Wisconsin. They allow stem cell research. They support human embryonic stem cell research. Well, states represented by South Dakota and a few in mid-US hugely oppose stem cell research. Therefore, in some of the states, you have a state-supported program. That means a lot of money from the budget of the government is used for stem cell research. Like we discussed, Irving Weissman was the one who pushed California to establish a stem cell research program. So if you're interested in that, you want to do you want to do your medical training or your PhD training in the field of stem cell research, go to California or go to Massachusetts, because they have good program. Your likelihood to get funded is much better than other states. Don't go to South Dakota. Those just, you want to get a chance. Because you can always get supported from the federal government, but if you also have local support, that's a lot better. Unfortunately, in this country, we don't have in our states there is no formal policy, and we don't have a stem cell program. Our emphasis in scientific research is cancer research. So we have a very good cancer research program called CPRIT. So your question, please. So somebody tried to ask a question?
Yeah, so
Okay.
so in the map, so is the map like according to 2011?
Yeah, it is.
So how did the perspective change till like 10 years after? Did it become a little loose-ended?
Oh, well, I guess most states will remain the same, but if you want to know the up-to-date information, we need to check for individual states. I guess the information, this is a new book, so apparently they took the most updated information from somebody's review paper. So that review paper is from Rice University study called Stem Cells and Biomedical Research in Texas. But as you see, it is sort of outdated.
Yeah, and
the updated information.
Okay. And you said like if you like use the ones before, like the stem cells before developed before 2001, you can be funded from the government. But if you use the ones that was developed later, you have to go through privates. But how much money are we talking about?
Well, research is very costly. So like me, if I have a project, if I will be able to convince the study section is experts, 20, 30 experts in a certain area. They will gather, they will have a meeting, discussing like 50 or so different proposals. If they agree that your proposal is valuable, that will give some insight into curing some disease or mechanisms of some regeneration, they can give you like $2 million to do it. So that's the $2 million is only for like 5 years. So for continuous research in a certain, just a very narrow area that you're interested in, we're talking about big bucks.
Because I was studying for my last exam in biochemistry, and then like there was a section in like how much money it takes to develop a new drug, and it's like 500 to like $2,000 million to develop just one single drug.
Of course.
So I was just trying to compare it, like how costly it is.
Yeah. That like I said, it's even if you do research in a very, very narrow area, without talking about developing a drug, it's just like you're studying a pathway in limb regeneration. You're using frogs, just a single pathway. That could cost several $1 million. And I remember you're only one of the scientists who do that kind of research. There could be 100 in US doing the same research. So research is very costly.
Yeah. All right, thank you.
You're welcome. So I guess when Barack Obama was president, he lifted that policy again. He said that now he's going to support human embryonic stem cell research, but I didn't know that Donald Trump did anything. So probably we are still at the Obama stage policy. So we can do stem cell research. Yeah, but that cutoff may still apply, I feel.
That's so
at least the last time when I write about human embryonic stem cell, when I checked the policy is still there. That was in Barack Obama's time. Yes, there's the about what kind of stem cell can you use. that 2001 cutoff still applies. But they do support stem cell research. Now, not only the government have some regulation over stem cell research, and because the huge promise that human stem cell research offers and other adult stem cell research offers, it's no wonder that a lot of companies start to exploit the opportunity. And unfortunately, the people they're trying to exploit is the people who suffer a lot because of some very severe diseases. And here I give you the result of a survey, like about people's opinion over stem cell research. So here is, if it's blue, light blue, that means the people say that they definitely would consider stem cell therapy as an option for their disease. If it's purplish, that says that they would not like to have a stem cell therapy as an option. So look at the vertical axis here. So that describes the severity of the disease. As you can see, with the severity of the disease moves up, then people start to become desperate, and then they turn to say that they would like to have stem cell therapy as an option for them, because when they have a terminal disease such as cancer, and the current therapy is not effective, and of course they're going to look for something else to try to save their lives. This is very natural and straightforward. Unfortunately, that gets exploited by the commercial companies. And at one time, it was very popular in countries like Thailand, Costa Rica, Mexico, Argentina, and to a lesser degree, China, India, and Russia. They have clinics offer stem cell treatment. So often, like I said, they take some stem cells from the same individual, and then after in vitro expansion, and then they put the stem cells back. They claim it is useful for treating quite a few diseases. The numbers of that scope is quite big, but in reality, in this country, I don't think we have any stem cell-based drug approved yet. So that says the scientific community and the companies have a very different view over the usefulness or the current status of stem cell therapy. And here I'll give you a number of how much they will treat, how much they will charge. Usually it is in the range of 5,000 to 40,000. But the problem is the treatments are unfunded. So they don't have scientific evidence to support that the treatment is effective. So they just take advantage of the people's desperation and then offer something they can't back it up, and that led to problems. Although in most occasions that injection will not do a lot of harm because it's the cell from the same individual and just expanded in vitro and injected back. Usually it is fine, but sometimes it can cause problem. Like here, it is an MRI scan of the brain of a little boy. This little boy he suffered from a neurodegenerative disorder, and he had a neuron stem cell injection in Russia. But you can see there, the arrow pointing to a tumor. So that eventually this patient had a tumor development in the brain. So a major challenge or problem with stem cell research is the tight connection between stem cells and cancer. So they can self-renew and then they can differentiate. That's the criteria of stem cells. But if something goes wrong, they lose the ability of differentiation, then their behavior will be quite like cancer cells. They will just self-renew, and then that will cause, you know, a growth, a new growth. If it's in critical place, like in the brain, that could be deadly. So that's why we need a lot of regulation over stem cell treatment. Please.
Yeah, so I have a quick question in the previous slide.
Mm-hmm.
So if you go there in the picture of the MRI scan,
Mm-hmm.
and there's a tumor and it had been treated with a stem cell. So my question is that sometimes in the MRI scan, if you have a tumor in the brain, it's rarely case that it didn't catch. So my question is that suppose if there is a tumor in the brain which is causing the body to paralyze one side, or couldn't connect or couldn't the brain, when the suppose if a brain couldn't send a signal to one side of the body, is that by using a stem cell, can we solve it, or can we like, so like this is my question, like.
Okay, so the boy had a neurodegenerative problem. That was the disease. It's not a cancer. So they tried to inject stem cells in the brain, into the brain of this boy, in hope that the stem cells will help the regeneration, because that disease was neurodegenerative. That means the neurons experiencing apoptosis or senescence, and then the neuron lose function gradually. So they feel like if you have stem cell injection, maybe it can restore some of the neurons and then therefore restore function. But unfortunately, restoring function didn't happen. A tumor was formed. The tumor was formed from the injected cell. They were able to prove that. It's the injected cell that caused the tumor formation in the boy. But of course, like you said, in the future, maybe we can inject the stem cells for the purpose of correcting, you know, the people get paralyzed. You may be able to help them, but we just don't have the evidence yet. At one of the lectures, I told you that you can put some nerves to an injury, a spinal cord injury, and put some olfactory bulb cells in there. You know, eventually it showed that the patient have a lot of improvement. That's the thing we need to be doing. You know, eventually to say whether we will be able to use stem cells for a certain condition or not.
Oh.
Yeah. It's not like we can just go ahead and just do anything we want. Not working like that. And so the therapy, stem cell therapy, has been done for recent years because many countries start to have regulations. You just can't, you know, just take some cells, no matter it's from the same individual or different individual, and then do the injection. You must prove that injection is effective. So the company just feel it's harmless. Now just do the injection and take the money. That's not, you know, that's immoral for most people. That's wrong, you know, to earn money in that way. But there are other companies who offer banking stem cells as a pay-for-service. That is also, you know, it's kind of similar to the companies that try to do stem cell research. In this case, they cause even less harm, because they are not injecting anything. They just take the stem cells. But you know, again, nobody knows whether those cells will be useful or not in the future. So basically, they are making money without a proper grant. Okay, so if you think that way, it's still wrong, but it is very popular these days. A lot of sources are exploited by companies such as umbilical cord. You know, you have a newborn, they offer you to take the blood from the umbilical cord, and then even infant teeth. infant teeth, like when everybody got 2 sets of teeth at year around 4 or 5, right? The first set of teeth will fall off, and then later on you have the permanent teeth. That's what happens. So a lot of companies are trying that opportunity. They will tell you, okay, now there's stem cells in there, in that teeth, in that tooth that just fell off, and now if you have a kit at home, you can put that into that kit, and then you send to us, and then we will process that tooth, and then eventually we keep that in a bank, in a tissue bank. And look at that price. So the kit itself is $60, and then the isolation is for a fee of $320. But the money call is the annual fee. They're going to charge $200 per year. Just imagine you start with from a little girl, a little boy, that's 4 or 5 year old, and now, so if you're going to pay the money, you're going to pay 80 years, 70 years. Imagine, that's a huge amount the companies will be able to collect. So that is actually very popular.
That's crazy.
Yeah, that's crazy, but that's what they offer in UK. That's actually very, very popular. That many years ago, that's many years ago, when my wife gave birth to my boys, that always get offered, like take the umbilical cord, then they do the extraction. Of course, they are going to collect the annual fee. The annual fee is the difficult part. It's a huge amount of dollars you have to spend. The problem is you don't know whether those will be useful. That's the problem. If it's proven to be useful in the future, and then it's legitimate, you will pay the money. It's as an insurance, right? But it's not the case. But then a lot of other companies are doing this. They know that it's easy to make money if you have a cosmetic product. So they say they have plant stem cell extract in their lotion. We haven't talked about plant stem cells. I don't know how those stem cells are similar to human stem cells, how the plant stem cells is superior in giving some extract, so that will help to make beauty product. I just don't know anything about that, but it is just absurd, in my opinion, to claim that. And then, of course, that's about regulations and the commercial companies trying to take advantage of the hope of the promise brought about by stem cells. And here I would like to point another direction. This is a mixed bag. This direction is a mixed bag because apparently not a lot of people will feel this is immoral. This have strength and weakness, have good and bad. So you can use stem cell technologies to clone animals or bring back extinct animals. Like here on the top, what's shown is the first clone dog family. So later on you will see it is very, very strange. It's a strange thing because the pup produced through somatic cell nuclear transfer was done by a Korean scientist. The dog is named Snuppy, but it was very, very difficult because Snuppy was the only survivor from more than 1,000 transplanted embryos. So they did a lot of those somatic cell nuclear transfer, but after 1,000 implanted embryos, only one survived. That's Snuppy. It was very difficult, but later on it became easier. From Snuppy, they take a somatic cell nucleus and then do nuclear transfer, and they got this family. They got quite a few dogs out of it. So I don't know the reason. Maybe their technique just become better. So it is debatable whether we should be doing this. Like when the dog's dying, should you to do a somatic cell nuclear transfer so that you can have a very similar dog. But to some people, they feel like it's going to be very difficult for them to to say bye to their dog. So this offers the opportunity for them to have some relief, emotional relief. So that's debatable whether we should be doing that. I like mammoth, and in bottom A, that's I proudly say that I visited this museum. It is Royal BC Museum. I believe it's on Victoria Island of Vancouver, and they have this very, very cool museum that they have artifact and a painting mixed together. So this mammoth is a it's an artifact there, but they have painting in the background and some setup in the foreground, and you almost can't tell that setup is unreal. You feel like, wow, it is a huge space there. You have a mammoth, but actually it's a combination of setup and painting and making look really nice. If you have a chance, pay a visit to that museum. It's a very unique one. But the thing here is, in 2014 in Russia, they digged out the remaining of a mammoth that is like 43,000 years old. But they were even able to discover blood in that remaining of the mammoth. When the blood is available, of course they can take the some lymphocyte and then have a nucleus available. So maybe there is somebody trying to do a somatic cell nuclear transfer and then implant that embryo to a elephant. Maybe we can bring back an extinct animal. Again, it's debatable, but I'm all for it. I feel like mammals are some really great species ever created, and their extinction was said to be because of human beings, our activities and hunting that make them to disappear not a long time ago. Then bring them back may not be a bad thing. And I guess this is close to the end. I just want to tell you how things are regulated. Drugs and stem cell treatment are regulated in this country, and probably you know this. So for a drug to go to clinic, it takes four phases. So phase 0 is basically what happens in labs, like in my lab, in somebody else's lab. We use animals to show that potential treatment is effective. We use cells, we use all kinds of things, just not human beings. So that is phase 0 and the development stage. But once you move to phase 1, you test potential therapy in humans. The phase 1 is about safety. It's nothing about efficacy. So in phase 1, you don't care about whether a drug or stem cell therapy is effective or not. You just care whether that will cause problems. That's what you care. Whether it will kill a person or whether it will cause some very severe side effect. That's what you care in phase 1. If you pass phase 1, you go to phase 2, then that is about efficacy, whether it is useful in treating the intended problem. And then in phase 3, it is both safety, efficacy, as well as dosing. So how is phase 3 different from phase 2? In phase 3, usually you compare that new therapy to existing therapies. So if your new therapy is less effective or more toxic, why will we need this new therapy? So in phase 3, it's usually larger scale compared to phase 2, and also you compare to other existing dominant therapies that will happen in phase 3. And eventually you have approval from FDA, but sometimes the whole process can be accelerated, like the COVID vaccine. Otherwise, the whole process could take more than 10 years or even longer than that, from the beginning to the end for a drug to get finally approved. But Sometimes you have this term compassionate use or right to try. You can skip all the phase 2, 3 and get a straightforward approval. That's called compassionate use, like some of the drugs during COVID. So as long as it is a drug already in the clinic treating other diseases, we know it is safe. So basically phase 1 has passed already. It is a safe drug. And now the doctors can decide, oh, just to use this drug for my patient because there's no other therapy. And this was proven to be safe for other problems. I will just try this on my patient. The doctor have the right to do it. They won't get into legal trouble if you have a ground to put an existing drug in compassionate use. So some other times that even compassionate use will have to be approved by the FDA as well. But as you can see, the companies who does stem cell tourism, they don't care about that. They just don't care about all the approval procedures that just jump to bedside directly. That is why it is bad. Now, finally, I would like to tell you some infamous people who messed up the field. And this guy is called Hwang Woo-suk. He's a Korean scientist who claimed that he had established the human embryonic stem cell lines from clones. That is the method by somatic cell nuclear transfer. So basically, he claimed that he have somatic cell nuclei he put into oocyte, and he was able to get embryonic stem cells. He published 2 papers in 2004 and 2005, but later on, both were retracted. So he basically fabricated all the results. And he also have some ethical problems, like using patient eggs without consent. It's just to get somebody's eggs. He even got the eggs from the postdocs, research associates in the lab. It's just to ask them to donate. And those, under him, they also want to, I think for those, they're out of their passion toward stem cell research, they were willing to donate, but still, it's not ethical to have that kind of to practice. You must have the consent from somebody, who, like in a clinic, somebody is doing in vitro fertilization already, and then it's much easier and more natural to go that direction rather than asking somebody to donate eggs solely for research purpose, because other options are available. But interestingly, he's the one who did this snappy dog. So basically 2 years later, when he was, people know that he fabricated results. He published papers, and eventually nothing was true. But he abandoned his research in generating human embryonic stem cells. He started clone dog, and he was successful. He was the first to clone the dogs. So still, he still had a lot of patent about using somatic cell nuclear transfer. And nowadays, if you want to clone a dog, the price can go from 50,000 to 100,000. That's the ongoing price if you want to clone a dog. And lastly, I want to talk about this pair, and especially this lady, this Haruko Obokata. And she claimed that she can induce the pluripotent stem cells. That's the cells Yamanaka generated, right? Induce the pluripotent stem cells by simply dip the cells in acidic solution. Just put some somatic cells in acidic solution, and after a few days, those cells will induce the pluripotent stem cells. It was very astonishing. I kind of lived that part of history because I was in a meeting in Galveston. that was a collection of some of the best scientists in the stem cell field. We used to have a really, really good stem cell meeting in Galveston, Hotel Galvez. But later, because of the attendance, the number of attendance wasn't that great. They removed that meeting. Otherwise, every year we're going to have a really great gathering in Galveston. Some of the best scientists, I knew them from that venue. And then in 2014, in that meeting, everybody's talking about this new technology, so-called stimulus-triggered acquisition of pluripotency. And the paper was published in January. The meeting was in March, but only 2 months, everybody said we can't, reproduce what she claims. And everybody says like we must do something. We can't allow somebody to just jump out and say something that's false and without good scientific evidence. And then 3 or 4 months later, and her institute start to investigate her, and then eventually, found that she produced one of the biggest scheme in scientific research, and she's a person that you can't understand. She, when they have her PowerPoint, she have a PowerPoint presentation, or she used the PowerPoint to prepare her figure, eventually published in Nature, and the IT people was able to find that the same figure, the same image, was used for maybe 30 different purposes. It's the same image of embryonic stem cells, or I don't know what image was that, but she just keep on changing the title. And this time it's an embryonic stem cell, next time is an induced pluripotent stem cell, and next time is just something else. She just changed the names for the same figure, that shows you how, what kind of person she is. It's just terrible. How can you do that? And then she was, her degree was taken away. Although her PhD degree has nothing to do with this discovery, but they found that even her degree, everything was made up. So eventually, her PhD degree was removed by the institute where she graduated, and then that also caused this co-author and mentor of her to hang himself, in the adjacent building. So after that investigation is concluded, a lot of scientists were affected. I know a very good scientist. I used to get reagent from him. It's a very respected scientist. I can claim that all his reagent is good reagent. It's valuable resources for us. And that professor, because he had a name in the same paper, and he had to resign from that job. And then he went to a much lesser, smaller university in Japan, because he's not involved in fabricating data, but he's a co-author. Just because of that, he was kind of forced to leave his position. This is something that happened in the history of stem cell research. It also tests from the other side that how powerful stem cell research could be. The people are just trying to jump on the wagon because there's a lot of hope in there. So if you jump on it, you can make an easy fame. But I guess most of us don't agree. We should do careful work instead of taking a shortcut, especially when it is so immoral. Okay, that's the end of today's lecture. So Wednesday, I'm going to have an overview, and hopefully I will be able to give you a good preparation for your final exam.
Thank you, Professor.
You're welcome.
Sir, it was an interesting discussion.
Professor, I have a question.
Sure.
For the clone dog family, is that a different method from like the Dolly the sheep?
It's exactly the same.
So there's still that drawback of like a shortened lifespan and all that?
Well, I think later on, when people start to do more and more, I don't think that's a problem now.
Oh, okay.
Yeah, the dotted sheep is just it's just one sample there.
Oh, okay.
So when they did more and more in cow, I believe there are more than 30 species, or even more than that. Different species has been cloned by using somatic cell nuclear transfer. So a lot of experiences, techniques are improving. So in the best hand, you barely see anything.
Gotcha. Thank you.
You're welcome.